Welcome everybody to the Sharp Minds lecture hosted by the Fleet Science Center. My name is Andrea Decker. I am the Adult Programs Manager at the Fleet Science Center. And every month I have the pleasure of hosting the Sharp Minds lecture series that normally, under normal circumstances, which we do not find ourselves in right now, happen at the Fleet Science Center on the first Monday of each month in our beautiful Haikov Dome Theater. At 10 o'clock in the morning. It's included with admission, and you also get to see one of our classic movies right after the lecture. But while we can't host it in person as a live event, we're super excited that we can still bring this talk to you online via Zoom. So that's why we're here tonight. If you missed the introduction slides, we have another Sharp Minds lecture coming up next month in June. It's on June 8th. That's going to be on women pioneers in space science with local space historian Gideon Marcus. He actually joined us for another space talk last year and was quite an audience favorite, so we're excited to bring him back. The Fleet also offers other science talks. We have our Hudson Science series that's online, and we're excited to have 2 more talks coming up that are focused on COVID and the efforts that are being done in San Diego to help combat the COVID crisis. In the last talk, we looked at the development of vaccine. The next one will look at finding treatments. And the last one is going to look at finding antibodies. All of the work is done locally by local scientists, and we're really excited to feature them and how they help battle the crisis that we found ourselves in. If you enjoyed this talk, which I hope you will, then please consider donating to the Fleet Science Center so we are able to keep this online engagement going until we're able to open our doors again to the public. I will put a donation slide up at the end of the talk with a link, so if you would like to support us, we would greatly appreciate it. And I think that covers my intro. I have the feeling I'm forgetting something. It'll come to me. The Q&A section. So normally when we host this live, we have the speaker give a presentation of about 45 minutes, and then we do Q&A with the speaker. We're going to do that sort of the same, but Dr. Marshall said, because she's kind of not jumping around, but she's got a couple of topics that she would like to hit, then we're both going to keep an eye on questions as they come in. So if you have questions, you don't have to hold back until the end of the talk. You can write it in the little Q&A section function that you see. It says Q&A has 2 speech bubbles. So just click on that. It allows you to type in your question and then hit send or submit, one of those. And then both Dr. Marshall and I will see it, and we will take care to answer as many questions as we can. There might be a chance that we're not getting to all of them. So if you have a specific questions that we didn't get to, but that you would really like to have answered, please email me at adultprograms@rhfleet.org, and I'll be sure to get with Dr. Marshall to get those questions answered for you in case we don't get to your important questions in this talk. All right, with that, I would like to turn it over to Dr. Marshall for this presentation.
Great, thank you so much. I'm excited to be here in my home with you, though. I will share my presentation. Okay, so yeah, it's wonderful to be able to share my work with you and the work of others. I'll be covering a few different stories today from a variety of people that have been in my lab, past and present. And we did actually a Suds and Science version of this a few months ago before everything shut down. And it was really a joy for me to see that people were really curious and interested. So I hope that you find this intriguing and fun, and I welcome any questions. I'll do my best to get to them during the presentation, but if not, I will definitely try to see them in the end. So today I'm going to talk to you about force sensing, so how your body responds to mechanical cues in the environment. And I think that when I tell people that initially, they don't immediately think of why mechanical force is so important in our body. And so one thing I want you to come away with after this is actually just understanding like all of the variety of ways that your body uses different kinds of physical force to function. And so I'm going to start by telling you about my lab. We are the Pataputian lab. I'm a postdoc, which if you're unfamiliar with sort of the course of training in science, that means I finished my graduate school work and now I'm sort of semi-intellectually independent, but I'm in the lab of Ardem Pataputian. So it's a really fun place to be because we get to just explore our curiosity. And you can see this is a great picture because it's actually a bunch of lab members, past and present, many of whom have worked on the stories that I'll tell you about today. But what I really love is this map that was put together by our administrative assistant, Angela, that shows the birthplaces of all the people that have worked in lab, because science is an international pursuit, and that makes it really fun. And there are a great group of people. We're housed in the neuroscience department of Scripps Research. And of course, right now, I miss them all a lot, but we're still trying to get together in socially distanced ways. So I wanted to start by telling you about the level at which we work. So I think when people think of biomedical research, they really think of this top of the pyramid, which is the patient relevance. You know, everyone wants to know that biomedical research will pay off and help people. And I agree that this is incredibly important. But the reason I wanted to show you this is to tell you that what becomes relevant for patients actually sits on a huge pyramid of other work that might not seem directly relevant. So you have clinical trials and preclinical research, which I think everyone has all heard of. But really, the foundation of everything that serves patients is basic science research. And the reason I feel that it's important to talk about this is because, you know, when you hear about basic science research, what that really means is figuring out, you know, how cells function or how different molecules function within cells. And it doesn't always capture people's attention as being immediately relevant to patients. But in fact, you cannot have the top of the pyramid without a really large base of knowledge, because you really never know what you're going to discover at the basic level that will end up helping patients or supporting the, you know, translational work later into the clinic. And so I just want to put out a plug for basic science research, because sometimes some of the most unexpected discoveries end up being really helpful later on. And so really this level of research is just asking, how do things work? How does our body work? And in the case of my lab, you know, how do our mechanical senses work? And so I'm going to start out with just talking about how you sense the world. And I think everyone's probably familiar with the fact that your nervous system is responsible for doing all of your sensing. In fact, your brain is what's responsible for perception. And so this is what takes in all of the information and forms a cohesive narrative about what's happening in your world. And it's really the peripheral nervous system, which are all the nerves spread out throughout the rest of your body, that do the sensation. And so these nerves are taking in information from the world, and they're specialized to do that. And then they send it back to the brain. And so really I'll be talking about the peripheral nervous system, this sensation side of things. And what's required for sensation are usually specialized organs, and then within those organs, specialized cells, and within those cells, even more specialized sensory molecules. And so I'll tell you a little bit more about those. But just to give you an example, we all are very familiar with the eye as a sensory organ. We know that when photons hit your eye, it goes all the way to the back of your eye. And if I expand out the back of your eye, you would see the retina. Obviously, this is not an accurate drawing, and you can ignore all the different cell types. The retina is very complex. It's a really amazing structure. But one thing to know is that these cells in the front are neurons, and they're what send the information to the brain. But since we're talking about sensation today, the sensory cells are at the back, and these are called photoreceptors. So these are the specialized cells that take in light and convert light into a signal. So the way that happens is, if I were to zoom in on this really tiny edge of one single cell, you would see this really complex cascade. And I don't want you to worry about any of that, but this is called the phototransduction cascade. And so what this means is photo is light, and transduction is how you convert a physical stimulus into a cellular signal. And so all this means is something that takes light and makes it a cellular signal. And really the exciting protein that does this is called rhodopsin. And this was discovered in 1876. And so you see it as a little, it looks like a little column sitting in the cell membrane. And the reason rhodopsin is the key here is because it's the protein that actually takes the photon, and then the photon changes it. And that's what starts this whole signaling cascade called vision. And just as a reminder, because I'm going to be talking about proteins and stuff, rhodopsin is a protein, but it's encoded by a gene. And so just to get everyone on the same page about this, DNA contains genes, and genes are the blueprint to make proteins. And proteins are the workforce of your body. So these are enzymes, and there are structural proteins like keratin in your skin and hair that give form. But today we'll be talking about sensors, and in particular ion channels that are specialized proteins to detect stimuli. And ion channels, again, if you only remember one thing about them, think of them like switches for your nervous system or for these specialized cells. They really switch cells on. And the way this happens is that ions flow through and they change the electrical properties of the cell. And then those electrical impulses send information to the brain, because really electrical impulses are the language of the nervous system. This is how your nervous system communicates. And so again, when I say ion channel, because I probably will many times, just think of it as like an on switch. And so next I wanted to talk to you about all of your senses. So when I say the senses, you probably think about these 5 senses. These are the Aristotelian senses, right? And we're all very familiar with all of these. And as I mentioned, light comes in through your eyes, a specialized organ, and then you sense light by phototransduction. But you also have chemical transduction that is evidenced by your sense of taste and smell. And then you have mechanical transduction or mechanotransduction. And we're familiar with this in the form of hearing and touch. But really, even this is such a limited view of what the senses are, because when we think about touch, say, we think of the skin as the specialized organ, it does so much more than just touch, because it has specialized sensors for gentle touch, say like the stroke of a feather versus a pinprick. And then of course it also does other things beyond mechanical sensing, like sensing thermal heat and cold. And each of these different modalities has specialized cells, and within those cells, specialized proteins that do the job. And so it's kind of amazing to think about all the diversity of sensors that your body has working all of the time. But even still, I'd say this is just a narrow view of what your body can do and what it's sensing, because this is exteroception, how we take in information from the external world. But we actually have an entire world inside of our bodies too. This is called interoception. And all of your major organs have sensory neurons sitting within them, and you might not even realize. And in fact, I think we all take these senses for granted. If you take a big breath, you can sense that your lungs are full. And you also know that if you're not getting enough oxygen, you need to breathe faster. And this is all done through senses inside your body that your brain might not even let you know that it's doing this. Simultaneously, your gastrointestinal tract, like your stomach, expands greatly when you eat. And then your entire GI tract is always mixing and churning and moving contents through. So there's a lot of mechanical force happening inside your gut. Your body knows exactly how fast your heart beats, and it knows when it needs to beat faster. And lastly, your muscles have sensors for stretch in them. And all of these organs function both they have chemosensors as well as mechanosensors. And a good example of this would be muscles. For instance, if you work out a lot, you know that your muscles burn, and this is a chemical sense. But you also know at any given moment where your arm is sitting. You don't even have to move. You can just think about it, and you know the position of your arm. And again, this is another thing that we really take for granted. This is the sense called proprioception, and it's just knowing where your limbs are in space. And the reason you know this is because you have special stretch sensors in your muscles that tell you where they are at any given moment. And so next time you walk, just remember that you have a sense of your body and your physical position. And that's something that's happening all the time without you even trying. And so our lab, as I mentioned before, is really interested in these mechanical senses. And just to compare, before we heard about phototransduction, this is called mechanotransduction because it's the force sensing channels. So when we talk about mechanotransduction, I think people think since all of the senses I've mentioned are very mammalian, we think this is something special about mammals, but actually everything senses force. It's actually really fundamental to living in any environment, really. So the first mechanosensors were discovered in bacteria, and those same sensors are also in plants. And so plants do have a sense of touch, and we all know this because of Venus flytraps. If you touch their sensory hairs, they snap shut. But all plants also know which direction gravity is pulling, and this is why roots grow in the proper direction. And so the sensors for these things in bacteria and plants had been known for a while, and we've also known what the mechanosensors were in worms and flies. But for a really long time, no one knew what the sensors were, the ion channels, those on switches were for mammals. And this is kind of crazy because I just went through all of these different physiological functions that mechanosensing has, but we just didn't know what that special sensor was, that on switch. And so this is what my lab was really interested in. And this actually precedes me, but we started asking this question a while back, which is how do mammals detect force? Because this is so critical to our daily lives. And this is a project that started, like I said, back in 2008 or 9 probably, when a postdoc like me, Bertrand, he started trying to figure out what the sensor was. And so I'm going to show you how he did this. He actually just took cells in a dish that were mechanosensitive. So that meant he would poke them. I'll show you here. I think it's very cute, actually. So he would go and poke cells. And what was special about these cells is because they were mechanosensors, whenever he poked them, you would get an ion channel current, which is what I mentioned before. These on switches would come on, and that's what conferred the mechanosensitivity, is there's a special ion channel that was opening every time he pushed them. So there was an on switch for detecting force. And so he went through a long list of genes and deleted them, hoping to see a cell, once the gene was deleted, that was no longer mechanosensitive. It actually took him quite a bit of effort. But on number 72, he discovered these ion channels called piezos. He named them piezos. And this was 2010. And I just want to point out, a reminder that rhodopsin, the equivalent for light or phototransduction, was discovered in the 1800s. So this just tells you that this field is actually pretty far behind in terms of understanding on a molecular level how things function. So this was a very exciting discovery. It opened up all of these new questions in the field about where piezo ion channels were functioning and how. And just to give you a more close-up, non-cartoon look at what we call piezo, my colleague, Kei Saotome, actually solved the molecular structure. And I'm not showing you this for any reason other than I just think it's really beautiful. But you can get some information just by looking at its shape. So you can see it has these 3 long curved arms that stretch out. And these arms actually are what's sitting in the cell membrane. So what happens, we think, is that when a cell gets pushed, those arms get pulled out in the membrane and actually open that critical ion channel to make the switch on. And so this is a really cool example of how form actually gives us a good idea about function. So there are 2 piezos, piezo 1 and piezo 2. I'll talk about them both a little bit. But piezo 1 is expressed in a lot of different tissues. We found it in the vasculature. The reason this might be important is you can see in this simulation, this is a simulation of blood flow through a bifurcated artery. And you can see there's all sorts of turbulence. And we think that these vascular endothelial cells experience a lot of shear stress because of all of this turbulence. So we found this protein doing a lot of unexpected things in tissues we weren't, really looking for them. So vascular endothelial cells, so that it's important for vascular development. It's also important for your lymph system to work. In fact, a mutation in this is associated with lymphedema, we found. And it's also in red blood cells. And this was a really surprising story because, again, we never really expected mechanosensing in these cells. And all of these are published work. We think that, the red blood cell story I won't talk about today, but it might confer some protection against malaria, which is a really amazing story. So I highly suggest you look it up if you're curious. But what we're concerned with here today is really the nervous system. And both piezo 1 and piezo 2 are in the nervous system, although in slightly different places. And so the nervous system, as I mentioned, is how you're sensing the world. So the way you sense external cues is through something called DRGs. And I'm just showing you the skin, a little diagram of the skin here. And as I mentioned, the skin has all of these different cell types to sense different stimuli, say like touch, deep pressure, pinprick, and heat or cold. And so you can see in this cartoon all of these different little endings. And these are the endings of the cell, but the actual cell body, which really look like the cells you learned about in biology class, sit all the way back at the spinal cord. So I'm going to show you just a little cartoon cross section of a spinal cord. And you can see this is what's called a dorsal root ganglion. A ganglion is just a collection of cells. And so you have a bunch of these little cells that live in this ganglion. And they're actually very long cells because they send projections to all of these different places, like the skin or like your organs. And so this DRGs are what sense outside world. You also have other clusters of neurons that send projections to your inside organs. And this goes through the vagus nerve, which is a really long winding nerve that, like I said, hits most of the major organs of your body. And so I wanted to show you a picture of what these clusters of sensory neurons look like. This is a picture I took a while ago. This is actually a bat DRG. And the reason I like showing this is because it gives you an idea of how much variation there is in these cells. You can see the cell bodies are sort of sometimes round, sometimes a little smushed. And in the middle, there's a nucleus. And here, they're all different colors because I've stained them for different proteins. So it just shows you how diverse they are. And they're also different sizes. And at the top, you can see some of the tracts that go, and actually, these are the tracts that are going out to various places in the body. And so this just gives you an idea of the diversity and beauty of these neurons. So we wanted to know, what role does piezo2 play in mechanosensation? Because it turns out it's actually really highly expressed in these ganglia. So I have a question. And someone had asked if the vagus nerve is damaged, can it heal? And it's a good question that I can't answer, very completely because it really depends on the kind of damage. But one thing I will say is that internal organ nerve damage is, I think, not appreciated as much because you're not always consciously sensing your internal organs. That means that some problems you might have, you might not directly associate with nerve damage inside your body. But we know that people with diabetes experience neuropathy, and that neuropathy is not limited to your skin neurons. In fact, it could be in internal organs as well, and that certainly affects the function of those organs. At this point, it's not very easy to regrow nerves in humans, unfortunately. So, okay. We wanted to know what role piezo2 plays in mechanosensation because, as I mentioned, piezo2 is the one that's in the DRGs. And what was really incredible is as soon as we undertook this project, they found very quickly, actually, that piezo2 is responsible for the sense of touch and responsible for that sense of proprioception that I mentioned before, which is knowing where your limbs are in space. And this is just an example of what happens on the left in black when you stretch a muscle. And you see these really big spikes of activity. And these are the electrical spikes or impulses that I mentioned before. And these, again, are how the neurons are actually communicating the muscle stretch to the brain. But if we actually use genetic tools to remove this piezo2 protein from mice, on the right in red, you see that these nerves are silent. You can stretch a muscle and you don't actually get any real response anymore. And so this was really exciting for us because both touch and proprioception were relatively unknown from a molecular standpoint. No one knew how they functioned. And so this was the first clue anyone had to understand the sensors that really, allow you to sense these stimuli. But what was especially exciting for us was in 2016, our colleague, Alexander Chesler at the NIH, had identified humans who had, lost function of piezo2. So these piezo2 deficient humans, grew up not knowing what was actually wrong, but they had a lot of difficulties. And so when Karsten Bonneman at the NIH, along with Alex Chesler, actually got in touch with them, these subjects were incredibly gracious and giving of their time. And so they actually got to find out so much more about what this protein is doing. So these people have no sense of touch or vibration. And you think that sounds like a really, huge loss, but they've grown up without it. So it's not something that they went and could tell their doctor, I don't have a sense of touch. They don't know what that feels like. And in fact, there's one sense of touch they still have, which is a gentle stroking on hairy skin they can still detect. And so that tells you that it's actually not depending on piezo function. But they also lack proprioception. So they've gone through the world not having any sense of where their limbs are in space. And I wanted to show you an example of what this looks like. This is a published with full consent from this piezo 2 deficient subject. And you can see that she's asked to do something quite simple. It would be simple for us, which is with her eyes closed, can she just walk in a straight line? And she has a harness to protect her. But you can see that she has a lot of difficulty simply walking forward. And she can walk, but what she's done, which is actually, again, incredible, is she's learned how to compensate for her loss of proprioception by using visual cues. So she actually has to physically look to know where her limbs are in space, because her body is blind to her sensation, which is actually it was a remarkable discovery. And these patients are so incredible because they've given their time. They've really helped us understand a lot more about how mechanical sensing works in the body. And so they've also opened up a new area of study into certain pain conditions. So one type of pain, there's actually many different kinds of pain, as we might all be aware. One kind is called mechanical allodynia. And all that means is when something that is not normally painful, say a gentle touch or a stroke, becomes painful. And so I think we're all most familiar with this, maybe in the context of sunburn. We've all been sunburned, and even just putting on clothes hurts. And that's mechanical allodynia. But a sunburn is very brief. And unfortunately, for people who have neuropathic pain, this condition can be chronic, where everything that touches them hurts all the time. And as you might imagine, this is a terrible condition to have. And for a long time, we haven't exactly known how this sense was conveyed in the body. But we used animal models to figure out that mice who lack piezo 2, and these are similar mice to the ones I showed you where their muscles don't respond to stretch anymore, they also don't develop mechanical allodynia. And the way we do this is by using capsaicin, which is another, it's the hot ingredient in chili peppers. I think we're all familiar with maybe having cut a chili pepper and then have this terrible burning. So that's a good way to induce mechanical allodynia that's very brief. So we found that mice actually lack mechanical allodynia. And then Alex and Karsten Bonneman at the NIH were able to figure if this is also true in humans. And one reason this is very important is because you might know that pain medications are really limited. And one of the reasons is many things that we find in mice don't always translate to humans. So what Alex and Karsten's group did is they asked the patients, obviously this was all fully consented, but they asked the patients after they applied just a control fluid and a capsaicin fluid to different patches on their arm. The person did not know where it was applied. They waited, and of course, you and I know that when you apply capsaicin, your arm gets red. And so they had a control to make sure the capsaicin was working in these patients. And so you can see on the top, both the top and bottom, after the capsaicin, you see this Doppler shows this inflamed area. So that means the capsaicin was working, and it should be painful. But then they touched them in both areas, A versus B, and asked which was more painful. And, all of us, I think, would know immediately that the place where the capsaicin was more painful. But when asked to choose, these patients chose at random because for them, it was no more painful than it had been before. So they couldn't tell which patch was more painful. And this was a really remarkable breakthrough. And of course, it has profound clinical implications because if you know what's actually transducing the pain, what's really there at the very first step, then maybe you could interfere in some way to help these patients. And I see that there's a question, so I'll try to, oh wait, no, I'll wait on that. So this was, again, really exciting because there had been some controversy as to whether allodynia was conveyed by touch neurons or pain neurons, because these are different. And so this really, I think, put that to rest. And now it gives us maybe a therapeutic target to follow in the future. So next I'm going to tell you about other mechanosensory functions, because again, I was telling you mostly about kind of external reception and how you sense the outside world, but people sometimes forget how much you have going on inside your own body. And so I'm going to tell you about a story on blood pressure sensing. You might not know it, but your body is always tuned in to your own blood pressure, even if you're not conscious of it or can't sense it. This story, I wanted to show a picture of Wei Zhang, and this is my boss Ardem on the right. Wei Zhang really pioneered this study and did a wonderful job. And Ardem is just a great mentor who supports us going into all of these different fields. Because as you imagine, we cover a really broad swath of physiology. So you might know that, we all, I think, have been to the doctor and had our blood pressure taken. This is really a critical vital function that every doctor always reads out because it's so important for your health. And it's an indicator of cardiovascular function. And so because it's so important, your body has a lot of different ways to control blood pressure. You can control it in a short-term system, and there's also a longer-term system that helps keep your blood pressure in check. Today, I'm going to talk to you just about this short-term system. And just to remind you why it's important to keep blood pressure down and keep it healthy and keep this system intact, when you have persistent high blood pressure or hypertension, it causes a number of problems. Because if you think about it, blood supply is important, again, for every organ to function. And so you can have an increased risk of stroke or heart attack, or even, kidney failure because of chronic hypertension. So this is why this system in your body is so important. I also wanted to show you this ad I found in a National Geographic from 1949. It turns out we've known about hypertension and blood pressure control for a really long time, and we've known it's really important. And this ad just cracked me up. It's paid for by the Metropolitan Life Insurance Company. I guess a life insurance, they want you to stay healthy, and this is why they're educating you about high blood pressure. But I wanted to cue you into this, my favorite part of this, which essentially says that patients with high blood pressure are advised to work and play at a slower pace. And this is very naive thinking. That's totally wrong, because now we know, it's been a few years and we've learned a lot. And in fact, exercise and staying active is the best thing you can do to keep your blood pressure in check. I just thought it was funny because even though, we've known about hypertension for a long time and we've known about blood pressure control for a long time, we've learned a lot since 1949. But even with all of that knowledge, in 2018, we still didn't know what the sensors were in neurons that conveyed your sense of blood pressure. And so I'm going to tell you about, this is called baroreflex, your sense. And so what this is it's a way to control your blood pressure that acts very rapidly. It's a negative feedback loop and it's homeostatic. And so what that word means is it's just your body keeping things in check. And so to describe this, you have sensory neurons. These sensory neurons come from your aortic arch and your carotid body. And so you can actually feel, if you feel your pulse in your neck, this is your carotid fork where these baroreceptors sit. And it's actually extra stretchy there. And that's how these sensors that sense your blood pressure actually function, is they sit in the wall of your aorta and your carotid. And each time your heart beats, they get stretched. And that's how they, if your blood pressure goes up, they sense a bigger stretch. And if it goes down, they sense a smaller stretch. So this is the sensory mechanism by which you control blood pressure. And just, this is always working. And to give you an example of how it works, if you were to rapidly stand up right now, your blood pressure would drop really fast. And this is not good because we all need blood to go to our heads. And so the way this baroreflex works is as soon as you stand up and it senses that drop in blood pressure, the motor neurons actually, it goes through a reflex arc and it tells your heart to speed up, and then it tells your vasculature to constrict. And so with your heart beating faster and your vasculature constricting, it keeps all the blood in your head. And that's actually why you don't faint when you stand up, I hope. And that's because your baroreflex is always working rapidly without you even realizing. And so people that have compromised baroreceptor function sometimes have arrhythmias, hypertension, and heart failure. So as I mentioned, this is an incredibly important system. And so I also wanted to tell you, I told you that these endings sit in your aortic arch and in your carotid. And the endings themselves of the neurons are really beautiful. So on the left is an old drawing of one of these flowery endings. And on the right is my picture that's a little bit zoomed out. And you can see that these flowery endings form a daisy chain right around the middle of that aorta or aortic arch. And so they're really, primely tuned as a cuff to sense your blood pressure, which is incredible. So one way we study this is by using a drug. So this drug is called phenylephrine, and you don't have to worry about what it hits, but essentially all it does is it induces vasoconstriction. So as I mentioned before, it doesn't work directly on the heart. It works on your blood vessels. And as I mentioned, if you constrict your blood vessels, that raises your blood pressure. It keeps more blood centrally. So what we do is we can give phenylephrine, and I'm just going to show you a little, trace of what happens. The top is blood pressure. The middle is your systole, which is also a measure of blood pressure. And you can see that when phenylephrine is given, your blood pressure goes up. And at the bottom is heart rate. And you see this, dramatic decrease in heart rate to compensate. And this is the baroreflex in action. But if we remove both piezo 1 and 2, before, piezo 2 works on its own in some systems, we found in this one it requires both piezo 1 and 2. We see that baroreflex is totally gone in animals where we genetically modify them so that they don't have these channels. So this was, mind-blowing, because again, this was only in 2018 that we understood how this critical reflex functions. And it was so exciting to find out. In fact, at first Wei Zhang tried just piezo 1 and they were still fine. And then he tried just piezo 2 and they were still fine. And he was a little bit sad, but the system is so important that it uses both together. And I'm just gonna show you a graph. These animals that I might say this again, so I'll tell you that KO or DKO, they mean knockout. This is just a name for the genetic tool we use. So if we knock out those proteins, you can see that they also have high blood pressure. And this is exactly what we see in humans with this problem too. So this was a big discovery for us, and we're still curious about where else piezo ion channels could function. So I showed you today in little brief glimpses that these ion channels are important for touch, they're important for proprioception and certain kinds of pain, and baroreception or blood pressure sensing. But we have so many more questions because, as I mentioned, there's so many different mechanical senses, and two of the ones I'm most interested in are how does your stomach feel full? When you eat a huge meal, we all know that you have that sensation of stretch and fullness, and you can't possibly fit any more inside of you. And again, this is not understood. We don't know what those signals are that are conveying that sense of fullness to the brain. And I'm also really interested in bladder fullness. This is another sense that we really take for granted because knowing when our bladder is full helps us plan, right? We can kind of think to ourselves like, oh, I need to get to the bathroom in about 10 to 20 minutes. We can sort of plan ahead because we actually are sensing our bladder filling and we know when it's time to go. And the reason this is so important to study is because so many people have issues with urinary function. In fact, almost 70% of people over 40 will at some point have some sort of problem. And that's because there can be so many problems in the system. You can have overactive bladder, underactive bladder, or incontinence, a number of other things. And given that we do, urination is something we do many times per day, when you have issues with it, it becomes a huge quality of life problem. And so again, this is something that we take for granted, we don't really talk about very much, but is a really critical sense to maintain our quality of life. So I just wanted to thank everyone in my lab. I have such a great group of lab mates that I work with. And as I mentioned before, most of the stories that I presented today were really spearheaded by other people in the lab. My stories are still kind of new and they're not out yet, but I would love to come talk to you about them more at another time. So the people who contributed are bolded, but you can see we have a really big lab. I also wanted to really thank the Chesler lab. Alex Chesler is a great collaborator and a really great scientist. And the Chesler lab and the Bonneman lab both have been incredibly generous and they share their data and they're the ones who work with these piezo 2 deficient humans. And then the Wilkinson lab actually collaborated with us to do the muscle stretch experiments. So these are the Twitter handles of myself and my boss. My boss is very entertaining on Twitter. So yeah, I would love to hear your questions, and you can follow us for more science.
Wonderful. Thank you so much, Dr. Marshall. This is great.
Good. I'm glad you liked it. Should I stop sharing or continue?
Yeah, if you wouldn't mind stop sharing, then I can throw up another slide in a minute. But I want to get to questions first, actually, because we have a good amount of time for questions, which I love. And I have the feeling that many people might be writing questions right now. So to get us started, I'm going to ask a question. You mentioned the bladder. I find it so interesting, and I would love to understand how that works. So when I'm standing and my bladder feels full, I feel it. But then I lay down or sit down and it feels completely different.
Oh,
so
that's interesting.
how does that play into it?
Yeah, well, I think the fact that we are bipedal and we stand up, certainly that weight actually helps you feel the sense of fullness. And one funny anecdote I have is astronauts. You might have heard this, but astronauts can't actually sense when they need to go to the bathroom very well, and it's a big problem for them. You know, there's all sorts of contraptions that they need to use being in space to actually go to the bathroom, but it can be really dangerous, because if you think about what a liquid does in space is it clings to edges, right? It doesn't sit with gravity. And because bladder stretch is so important, and that kind of fullness and weight of the bladder is so important, what happens with astronauts in space in microgravity is that the urine actually coats the inner edge of the bladder. It doesn't fill up from the bottom. And so what happens is the only time they start to feel full, it's already full. It's filling from the outside in, which is not what you want. So it's actually, that's a good way to tell that, gravity actually really does matter for our sense of, fullness and that weight of the bladder.
Interesting. I had no idea. Thank you. And I see that the question has come in. Oh, it's actually a comment. It says, thank you. This was very cool.
Oh, good. I'm glad.
You're very welcome. We're glad you enjoyed it.
While people ask questions, can I make another little basic science plug?
Please, yes.
I have it on my presentation. I wasn't sure if there would be time for it, but I wanted to show really briefly an experiment that Wei Zhang did in the baroreceptor study, where he uses light to activate the baroreflex. The reason we did this is because someone could say, oh, piezos are gone. What if the neurons are just broken? Like, you say there's no baroreflex. What if they're just not working? So we wanted to make sure the neurons were still there and healthy. And so he could express a special protein in these cells that was light sensitive, and again, acted as one of those switches, an ion channel. And it's, then all he does is shine light on the neurons. And you can see that if it's a regular mouse that's not expressing this, nothing happens. If he shines the light in the wrong place, which is gray, nothing happens. But he can shine a light on these baroreceptors and get this really, big change in blood pressure and in heart rate, which is really cool to us because it just tells us that the neurons work and the system is all in place in these animals. They just don't have the baroreflex. And the reason I wanted to show this, if I had time, is because the way this works is something called optogenetics, and it uses a protein called channel rhodopsin. And remember, I told you about rhodopsin before, and this is channel rhodopsin, which is an ion channel that responds to light. And it has become one of the most important tools we have in neuroscience, because we can switch neurons on and off at our will by just shining light at them. It's super cool. And the only reason we know about this protein is just because of really basic science, where, in the 80s, someone was asking, how do algae sense light? And that's how they found this protein. And this is just an example of algae. These are single-celled organisms that, in this case, they travel away from light. I thought it was a cool video. And so the reason I wanted to show this is just because, if you were to talk to someone about, oh, I study how algae sense light, you might not think that's so relevant. But actually, it has become one of the most important tools in neuroscience in understanding, how neurons work. And so I just thought that was a cool example of how something really unexpected in basic science comes to play later. So that was my little soapbox for funding basic science. Are you still there, by the way? Did I lose you? Are you still there, Andrea? I might have lost Andrea. That's too bad. Let me text her. I'll try to answer questions and see if she comes in. Oh, she has internet issues, she said, and she will be back in a second. So if you have questions, I can actually see them. But I don't actually see any active ones right now. So please ask questions. I'm just here waiting on you. Oh, someone says wait. Yeah, the other example, I'll just talk for a minute about how unexpectedly helpful basic science can be, is CRISPR. I think everyone's probably heard about CRISPR in the news. And this is something that the only reason we know about this incredibly powerful gene modification technology is because someone was interested in the question, how do bacteria fight off viruses? And that's how CRISPR was discovered. And that's what has led to now this huge change in how we could modify genes in the future going forward, all because someone studied bacterial immune system, which is really cool. Oh, Maggie.
Say I'm back.
Yes, thank you for coming back. I was nervous.
Sorry about that. Sometimes it just completely just dies on me.
Terrible. So it looks like Maggie wants to ask a question.
Perfect. Let me see if you can do it by audio. Hold on. So, Maggie, give it a try.
Okay, thank you. I once attended a meditation course, and one of the activities was to scan our bodies, right? So, and the teacher, it was all recorded, and he just kept saying, you know, look for any sensation that you can. And it wasn't, I didn't feel like it was a problem for me. I mean, I could feel sensation all over the place, but depending on where I would put my attention too, because you can forget that you have a hand during the middle of the day. You can forget that you have these things, but if you give some attention, it's like, oh yeah, it's there. So, but I had given a ride to somebody, and when we were coming back, he was saying that he didn't understand what this person was talking about. And I was mystified by that as well. And it wasn't till later that I thought, I wish I had just said to him, you know, well, how do you know you have a leg? How do you know you have an arm? You know, like, don't you know that can't you feel that at some point? But it made me, as you were talking, I was wondering if he did have some kind of defect in his proprioceptive system.
Well, I mean, I think if you were truly insensitive and truly had no proprioception,
he would know by now.
Yeah, he would know. I mean, he wouldn't be at a yoga retreat, I think. But certainly people have different sensitivities, right? I mean, and certain people are really tuned in to their interoceptive senses.
Yeah.
And some people aren't. Like, I actually feel like I'd be very bad at sensing my heartbeat, but this is something that, I mean, if I exercise, I can feel it, but this is something that certain people are very tuned into. And I think part of that is, like you said, it's just attention, and it's also practicing that attention. And one thing I find really fascinating about this area, and this is something that I want to study, you know, far in the future, is understanding how that attention to your bodily senses actually affects your mental state. So there's this theory called the James Lange theory of emotion, which says that contrary to sort of your, you know, typical intuition, which says that, you know, if you feel scared, your heart starts beating fast. It says the opposite, which was really that you sense your heart beating fast and it makes you scared and it causes anxiety. And I've heard this from many people who have anxiety attacks, that it's really that it's like their body is acting without them, and that causes them anxiety. They get hot, they get, you know, they can feel their heart racing, that this causes the sense of fear and anxiety. And so I really wanted to understand sort of the bottom up direction of this, right? Because we know your brain influences a lot about your body, but I really want to understand how the senses coming in from your body influence your brain in different ways. And, you know, the opposite of this is depression, where people report anhedonia, like nothing feels good to them anymore. And in fact, it's sort of like a lack of feeling. And so I'm really curious about how all of these senses inside your body feed into your mental state. That's a lot farther away.
I've I've also heard or read that anyway, that they found that you think often that you decide to do something and then you do it, but they found by different kinds of brain scans and everything that often the inclination has actually already occurred, and then the person thinks that they're choosing it. So do you think, is that kind of related, do you think, to
it's sort of just generally related to how the nervous system functions. I mean, we all live in the past, right? Because senses take time. They take time to get from the outside to your inside. And certainly your brain has to work before it sends signals out, right? And so the same applies there.
Right. But it would also seem too, with what the situation you had talked about the anxiety, is that whatever situation it is probably has some, I just assumed right away that it was like it had a subconscious thing where there was a fear system that was ignited by a previous, event or something that's kind of in the subconscious, and then that comes out, and then you feel those things. But
yeah, it could be. There's definitely a lot.
You want to see if that's what it really is.
Well, I mean, there's definitely a lot happening under the surface, right? A lot of these senses that I was talking about, they like baroreception. You are not at all aware when your blood pressure changes, right? So some things, it's almost like your brain doesn't need to clue you into, but other things certainly. Oh, it looks like we lost Andrea. But other things, it does. And so it's more that I think that we know that we react if we feel something on our hand. If we have an itch or something, we scratch it, right? And that's a reaction to an external stimulus. We know a lot less about how we react to internal stimuli. A lot of, when you think about internal, gut sensing, for instance, generally, if people are sensing their gut, it's not good. Generally, it's painful, right? And this is because, the reason you have the sense of itch on your skin is because you can do something about it. You can't itch inside, you can't scratch inside your body. So there's no sense of having a sense of itch inside of your body, but you do have pain, and pain is critically important to tell you when something goes wrong. And unfortunately for some people, that really critical function goes haywire, and it tells you you're in pain all the time, even when there's nothing acutely wrong with you. And so, I mean, I think understanding how all of these peripheral neurons work and inform your brain about what's happening in your body is just, understanding just from a level 1 perspective of how these function is really important. Yeah.
Sorry, Maggie, I'm thank you so much for your questions. I'm going to put you on mute again and see if there's other folks who would like to raise the hand, ask a question, or type it into the question section.
Looks pretty quiet. We still have 20 people hanging around, but oh, I can't hear you, Andrea. Yeah.
I'm still amazed about the question about the stomach and how you know you're full.
Oh, yeah.
Just because, I don't know if that's true or not, but they say the slower you eat, the better you sense whether you're full or not. Is that true?
I don't actually know about like sort of the time, but I think it's the longer you wait, obviously, the more likely you are to feel that sense of fullness. And so we think, there's actually a paper that came out not too long ago that suggests part of that fullness sense is in your intestines. For a long time, people have been studying, the stomach stretch aspect. And so, again, because we don't know the sensor, it's been really hard to study just from a pure standpoint. We know a lot about hormones and different hormones that make you feel full, like leptin. And so once those receptors were discovered, that opened up a whole field of understanding satiety and fullness. So I'm hoping that with, ion channels like piezos, we can do the same thing for mechanical stretch in the gut. So someone actually has a question. So the question is, any idea if piezo 2 has any effect on response to skeletal muscle damage? Oh, and so, it's a great question. If, do mice lacking piezo 2 have trouble with muscle regeneration? I don't think that's been answered yet. So one thing to note is that in the muscle itself, the muscle cells, I don't think piezo 2 is expressed. So that gene isn't expressed there. So we don't think the protein would be in the muscle cells, But, if the neurons, could have some effect, like a trophic effect on the muscle cells, then maybe. I don't think that's been studied yet, but someone should study it. It's a good question.
Perfect. We're coming slowly but surely to the end of the talk, but there's another question.
Oh yeah, so someone asked, do you know how leptin works without something mechanical? That's an important question. I didn't really make that distinction. So leptin is a hormone which is actually released into your body. It floats around in your bloodstream and gets to your brain, and that's a signal to tell your brain that you're full, or that your fat content, that you're happy, that your fat content is good. It's released by your fat cells. And so that's a hormone, and that is actually acting through chemotransduction. So leptin acts as a chemical to activate its receptor. And so instead of the receptor, or instead of the activation being through force, it's actually acting through a chemical signal. So it's just a different kind of signaling mechanism. And so that is known and that's understood. And like I said, once that receptor was discovered, it opened up a whole bunch of really interesting studies on how these different hormones affect feeding. And what's not been done yet is the equivalent with mechanical force, because they're both playing a role together.
Thank you so much. So I'm going to share my screen real quick. I'm going to see how I can make sure that you guys can still see us. I'm still learning a lot about how Zoom works, but I think that is the same for a lot of us who started doing online events.
Definitely.
But I want to make sure that I share this slide with you, just so you know what's coming up in events, just in case you missed the beginning slides. So we have another Sharp Minds lecture series coming up on June 8th, the Women Pioneers in Space and Science. And if you are interested in learning more about what the San Diego science community does to fight COVID, then we have talks coming up in May and in June. As I mentioned when we got started, Sharp Minds is normally a live event that we host in the Hyckoff Dome Theater. It happens on the first Monday of every month at 10 o'clock in the morning, open to everybody. But we do have a special deal for our guests 65 years of age and better. The event is $10, gets you into the Science Museum, gets you the lecture and a movie right after the lecture, but it's open to anybody. So if you find yourselves, when we get back to normal, if you find yourselves for looking for something to do on a Monday morning, then I hope you will join us live. And we have another question here, actually. We know that sleep can affect our weight, our attitudes, and so much of our waking lives. Has anyone looked at any connection between sleep and your research?
I love that question, because it's actually an area I totally want to study. And so what governs your sleep is circadian rhythms. Sorry if you hear my dog running around. Yeah, so circadian rhythms are what govern, differences between, say your dark phase or your sleep phase and the light phase. And there's a lot of reasons why you really don't need mechanosensing in certain senses, when you're asleep, let's say. And so there have been a few papers that have looked at how circadian rhythms affect expression of these piezo genes. It seems that there's differences maybe in the skin and some other areas, but that is still brand new and I think not understood at all. And because circadian rhythms seem to function in all sorts of cell types, it would not surprise me if these proteins are also being governed by circadian rhythms. But it's an awesome question, and I have the same question, so I think it's a great one.
Great, we have one last question for tonight. Someone commented that they have read that it's a myth that the stomach stretches, but it sounds like there is some reason to believe it does, or that there is a mechanical element. Is that what you're saying you want to study?
Yes, so the stomach does stretch enormously. So anytime you take in food, it actually expands the volume that it can accept by like 16 times. It's called gastric accommodation. So there's this massive expansion, and that's how you're able to take in food. So we know that it stretches actively. And then of course, if you eat a lot, it probably is stretched by the food as well. And it's been known for a long time that there are sensors in the stomach that detect this stretching. Now, what those sensors are doing in terms of influencing feeding is not understood nearly as well. And the reason we don't know is because we don't know what the sensor is. So if you don't know what the sensor is, you can't actually take it out to study how mechanical inputs affect feeding and fullness. So there's only sort of like kind of weak correlative studies. So yeah, that's exactly what I want to study is, you know, if we can find the sensor, which I'm hoping are piezos, and remove them, then we can learn a lot about how mechanical force influences feeding. And when I say mechanical force, I mean force in the stomach itself and also in the gut. Oh. So the next question, which I think is from my mom, is stomach stretching involved when I'm hungry and press on my stomach, my hunger subsides? Maybe so. I think that's very possible. Yeah. You know, it's interesting because when you have to go to the bathroom, if you push on your bladder, it hurts worse, right? You can feel that stretch increases. And so maybe something similar is at play. Your stomach always has something in it, fluid or some other stuff. So yeah, I can imagine if you're pushing on it and it goes away, maybe it's activating those stretch receptors and telling you that it's more full than it is. That could be the case. Oh, hi, Peter. Yeah, so yes. I'm glad you came, Peter. He's saying, I'm glad we didn't have to fight for the same parking spot. So Peter came to the Suds and Science event, and we did the thing in the road where we both wanted the same spot, but he still came and we both got parking. It was great.
Perfect. Thank you so very much, everybody, for joining us tonight. Dr. Marshall, thank you so much.
It was great to be here. I really enjoyed it.
Wonderful. Since nobody else can, just because we don't allow it, I'll give you the super applause for tonight. Thank you so very much.
Have a good night.
Thank you so much. Everybody, thank you so much. I hope you'll join us soon. I'm going to throw up the slide one more time, just in case you were looking for the events until everybody is logged off. There's also the link if you would like to support us. We would greatly appreciate it. If you like the event, please consider helping us make sure that we can bring these to San Diego in the future. And of course, we're looking forward to seeing you live again at some point. Thank you so much.
Bye.
See you all soon. Bye.